Hello students, in this presentation I am going to talk about viruses, their cultivation and reproduction. Cultivation means growing the viruses into the laboratory as well as into the industry, and reproduction means multiplication of viruses. Into the previous lecture we have already studied regarding the morphology and classification of viruses. So virus is obligate intracellular parasite which requires host cell for its growth. Because the virus do not have metabolic machinery, for example ribosomes, enzymes and many other materials or cellular components which are required for multiplication as well as we can say replication. So virus enters into any kind of host cell, that may be either animal cell, plant cell or microbial cell, and it uses the metabolic machinery of that particular host cell and likewise carry out its replication as well as the growth of virus when it takes place. The structure of virus involves it is only the cellular particle. It is not cellular actually viruses. Viruses is only composed of either DNA or RNA, which is surrounded by the protein coat that is called as capsid. Capsid is composed of monomers that is called as capsomers, which is mainly composed of different amino acids. The capsid may be surrounded by an envelope. Okay, the viruses which contains the envelope are called as envelope viruses, and which do not contain envelope are called as non-envelope viruses. On some of the enveloped viruses, there may be the spikes. With the help of spikes, viruses attach to many other surfaces as well as the other microorganisms, or we can say other host cells. So this is the morphology, and on the basis of various categories, the viruses can be classified. For example, on the basis of genetic material, viruses are classified as DNA viruses and RNA viruses. Single-stranded DNA viruses, double-stranded DNA viruses, single-stranded RNA, double-stranded RNA, likewise. Now, depending upon the host cell they infect, the viruses can be classified as bacterial viruses, then animal viruses and plant viruses. Okay, so likewise there was the classification, and third may be the depending upon the morphology. For example, helical virus, icosahedral viruses, polyhedral viruses, then bacteriophages. Bacteriophage means the bacterial viruses. Okay, so this much we have covered into the last lecture. In this presentation, mainly I am going to talk about COVID-19 virus, then cultivation and reproduction of viruses. So COVID-19, coronaviruses are a family of viruses. The 2019 novel coronavirus, or the severe acute respiratory syndrome coronavirus 2, as it is now called, is rapidly spreading from its origin in Wuhan city of Hubei province of China to the rest of the world. In India, there are totally or approximately 63 lakh cases, and the death cases are approximately 98,000. So this virus is rapidly spreading because it is contagious, that is mainly transferred from one human being to other human beings. So due to this transference, this virus is dangerous, we can say. If you look at the structure of coronavirus, these are enveloped positive sense RNA virus. Okay, so it is a kind of RNA virus whose length is about whose diameter is in between 60 nanometer to 140 nanometer, with spikes like projections on its surface, which gives crown-like appearance under the electron microscope. Hence it is known as coronavirus. The coronaviruses are enveloped viruses, that is means it is a kind of RNA virus. So this is about the structure of the virus. You can see into the image. So it is one of the enveloped virus which has the spikes which are red in color, means that is shown into the image. Then there is inside the inside material is RNA as well as nucleocapsid protein. Then there is envelope protein is there. Then membrane protein has been shown. Then there is an enzyme that is hemagglutinin esterase, but this enzyme is not responsible for replication. And there is the lipid bilayer membrane, and the outermost part of the COVID-19 virus is spike glycoprotein. Okay, so likewise, these are the structural components involved into the COVID-19 virus. The epidemiology and pathogenesis. Epidemiology means what is actually the cause of disease, and pathogenesis means in case of epidemiology, what is cause as well as how to control the disease, as well as in case of pathogenesis, what is actually the acuteness as well as chronicness of the diseases. All ages are susceptible, means this virus mainly attacks to the groups of all ages, whether it is children, kids, in adults as well as elder persons. The infection is transmitted through the large droplets generated during coughing and sneezing by symptomatic patients, but can also occur from asymptomatic people and before onset of symptoms. The study have been shown higher viral loads into the nasal cavity as compared to throat, with no difference in viral burden between symptomatic and asymptomatic people. Patients can be infectious for as long as the symptoms last and even on clinical recovery. Some people may act as super spreaders. These infected droplets can spread 1 to 2 meter and deposit on the surfaces. The virus can remain viable on surfaces for days in favorable atmospheric conditions, but are destroyed in less than a minute by common disinfectants like sodium hypochlorite, hydrogen peroxide, and ethyl alcohol. Okay, so these disinfectants are also called as sanitizers. So these sanitizers are playing very important role in prevention of the transmission of this coronavirus. The ethyl alcohol, that is about 65 to 90%, is very effective as a disinfectant. Then sodium hypochlorite, hydrogen peroxide, by using these disinfectants, various areas can be sanitized, and ethyl alcohol can be applied onto the hands as well as other body surfaces, so that that virus, means that coronavirus, is particularly destroyed with the help of these disinfectants or sanitizers. So the use of sanitizers is one of the important preventive way to coronavirus infection. The common symptoms including are fever, cough, sore throat, headache, fatigue, myalgia, and breathlessness. So all these are the symptoms related with coronaviruses. Means if the patients are symptomatic, then some of the symptoms would be shown. But if the patient is asymptomatic, means these clinical features or symptoms may not be visible. Okay. So likewise, the infection is acquired either by inhalation of these droplets or touching the surfaces contaminated by them, and then touching the nose, mouth, and ears, eyes. Okay. That's why do not touch to any surface. Okay. If we touched, then immediately apply the disinfectant or sanitizer. Okay. Do not touch our hands to the nose, mouth, as well as into the eyes. Okay. So the washing of hands after every 1 hour is mainly essential according to me. Washing the hands with the help of simple soap or washing the hands with Dettol hand wash is recommended by WHO. So these precautionary measures should be taken from these COVID-19 viruses. Then how this virus is diagnosed, means how this virus is identified. So there are various tests, diagnostic tests, which are mainly performed for identification or diagnosis of COVID-19. A suspected case is defined as one with fever, sore throat, and cough, who has the history of travel to many other areas or who came in contact with the patient of COVID-19. Okay. So those people are diagnosed for whom there are the symptoms, or those people are diagnosed who come in contact with the COVID-19 patient. Okay. However, the cases may be asymptomatic or even without fever. A suspect, or we can say a confirmed case, is a suspect case with a positive molecular test. Okay. Specific diagnosis is done by specific molecular tests on respiratory samples like throat swab, then nasopharyngeal swab, sputum sample, and many more. Virus may also be detected into the stool and in severe cases the blood. It must be remembered that the multiplex PCR, that is RT-PCR panel, currently is available, do not include the COVID-19. But nowadays the COVID-19 can be diagnosed with the help of RT-PCR. The appropriate sample has to be sent to the designated reference labs in India. For example, National Institute of Virology in Pune, means initially it was the samples were only sent to these national laboratories, but so number of labs are mainly available at district level as well as tehsil level, and from these labs the diagnosis of COVID-19 is mainly carried out. Let's see, this COVID-19 viral structure as well as the information regarding COVID-19 is not the part of syllabus. But as we are studying the microbiology, we should know about the structure as well as various epidemiology, pathogenesis, treatment, diagnosis as well as prevention of COVID-19 virus. In case of treatment, the treatment is especially supportive as well as symptomatic. The first step is to ensure adequate isolation to prevent transmission to other contacts, patients as well as healthcare workers. Okay, so whenever symptoms are shown by a particular person, then he must be isolated from other peoples to prevent the transmission to other peoples. Mild illness should be managed at home with counseling, about danger signs. The usual principles are maintaining hydration and nutrition and controlling fever and cough. So we must be hydrated throughout the day as well as night. We can use the steam, we can have the warm water throughout the day, okay, and maintaining of proper nutrition and controlling the fever and cough is also been managed at the home. Routine use of antibiotics and antivirals can be prescribed by the doctors. Then in hypoxic patients means face masks, then high flow nasal cannula or non-invasive ventilation is indicated. In some of the patients who have hypoxia, means deficiency of oxygen, for them the ventilator is required to correct the deficiency of the oxygen. Okay. Currently, number of drugs, antiviral drugs, are being used into the treatment of COVID-19. Those are Tamiflu, Fabiflu, then remdesivir also. Okay. Then other drugs or medicines which are used to treat fever, like paracetamol, then various painkillers can be used into the treatment of COVID-19. See, this treatment is only available which mainly corrects the symptoms. These treatments do not actually eradicate the virus. Please take a note of this. Only antiviral agents like remdesivir or other agents may be helpful, but they are not proven to be 100% effective to kill the coronavirus. Okay, so the recovery of the patient could be done after 14 days. The symptoms may not be shown by that particular patient, and from that again the test is carried out, swab test is carried out, and if it is found to be negative, then we can say the patient is free from COVID-19. Okay, so our immunity power is mainly essential to fight against this coronavirus, and the proper immunity can be had. We can have proper immunity with the help of proper nutrition, as well as regular yoga, as well as meditation and many more exercises. In case of prevention, since at this time there are no approved treatments for this infection, prevention is crucial. Several properties of this virus make prevention difficult, namely non-specific features of the disease, the infectivity, even before onset of symptoms into the incubation period, transmission from asymptomatic people, long incubation period, tropism for mucosal surfaces such as conjunctiva, then prolonged duration of illness and transmission even after clinical recovery. Isolation of the cases is particularly important, or home quarantine is also important. The ventilation at home should be good with sunlight to allow for destruction of virus. Patients should be asked to wear a simple surgical mask and practice cough hygiene. Not only the patients, but we the people who are the normal people should wear the mask to avoid or to prevent the transmission of this COVID-19. And frequent use of sanitizer as well as washing the hands with disinfectants as well as we can say antiseptics are equally important to prevent this kind of infection. So, this new virus outbreak has challenged the economic, medical, and public health infrastructure of the world, including India. Time alone will tell how the virus will impact our lives here in India. More so, future outbreaks of viruses and pathogens of zoonotic origin are likely to continue. Therefore, apart from controlling this outbreak, efforts should be made to devise comprehensive measure to prevent future outbreaks of zoonotic origin. Okay. Nowadays, number of researches are being going on. Number of vaccines are being developed. Clinical trials are being carried out. Yes, one of the day will come when we will have the very effective, we can say, vaccine to fight against this coronavirus infection. But up till that, we have to take care of ourselves, we have to take care of others, we have to take care of family, as well as we have to take care of our country. Okay. Then the curriculum part is the cultivation of viruses. How viruses are cultivated? Instead of infectious viruses, what is the reason to cultivate these viruses? The reason is that in order to study the viruses, we will have to cultivate them, because the microbiology is the study of microorganisms, and these microorganisms must be grown into the laboratory, and viruses are no exception. So viruses should be grown into the laboratory, and apart from that, the various viral vaccines are mainly produced. Viral vaccines are produced with the help of viruses. Okay, that's why due to this medical importance, the viruses are mainly required to be cultivated. Now there are various methods by which these viruses are cultivated or viruses are grown, like by using laboratory animals, then embryonated eggs and tissue culture. So these are the three methods which are mainly used for cultivation of these viruses. First is laboratory animals. It is one of the oldest method for cultivation of viruses. It was discovered in 1900 due to serious risks into these animals like monkeys, mice, guinea pigs, rabbits. This method is not that much widely used. Okay, the viruses are directly inoculated into these animals. They are mainly observed, they are mainly incubated, and then growth is indicated by death, disease, or visible lesions. But nowadays this technique is not used. Then next method is embryonated eggs. Embryonated eggs means fertile chicken eggs can be used for this purpose, because eggs offer various sites for inoculation for the viruses. Fertile chicken eggs are incubated for 5 to 12 days, can be inoculated through the shell aseptically. See, this is the structure of the egg. The outermost part of the egg, which is tough one, is called as the shell, and there are the pores onto the shell. So with the help of a needle, we can directly insert the viral suspension at various sites within the viruses, within the egg. Okay, the outermost part is the shell, then there is the air sac, then there is shell membrane, then there is albumin, and innermost part is the yolk sac. Okay, or we can say here that there is the developing embryo, and this egg mainly offers various sites for inoculation of the viruses. Okay, so uh, the chicken eggs are mainly incubated for 5 to 12 days, can be inoculated through the shell aseptically. Means the viral infection is inoculated throughout the shell aseptically. Aseptic technique must be followed for injecting the viral suspension throughout the shell into the egg. Then the opening is sealed with paraffin wax, and that egg is incubated at 36 degrees Celsius for the time required for the growth of virus. The duration and incubation may depend on the type of viruses and the route of inoculation. Viruses may kill the chick embryo or produce specific evidence of viral activity, such as production of pox on chorioallantoic membrane, as well as many more. Okay, so here we can see there are various membranes like chorioallantoic membrane, then amniotic fluid, then allantoic fluid, as well as there is a yolk sac. Okay, so in these cavities we can insert, means the viral infection, viral suspension is introduced, and likewise the growth of virus mainly takes place. Inoculation into allantoic cavity provides rich yield of influenza and some of the paramyxoviruses. Then inoculation into amniotic sac is employed for primary isolation of influenza virus. Yolk sac is mainly used for cultivation of some of the viruses like chlamydia and rickettsia. Yellow fever and rabies virus are other vaccines produced from chick embryo likewise. Okay, so it is one of the widely used method. So the advantages of this method is that the eggs are much simpler to handle than the animals. Eggs are very economical and they are easily available. They are clean and bacteriologically sterile. They do not need feeding and caging in case of animals. They do not have an immune mechanism like animals to counteract the virus infections. So the animals have the immunological mechanism which may fight against that particular virus. But eggs do not have immunological mechanism to counteract this virus infection, and chick embryo offers several sites for cultivation of viruses. So due to these advantages, the embryonated eggs are widely used for cultivation of viruses. Then there are certain disadvantages. Those are, some of the viruses do not show growth on primary inoculation into the eggs. Okay, then slight amount of bacterial contamination into the inoculum may kill the embryo. Okay, and eggs may be contaminated with mycoplasma and latent fowl viruses, which may interfere with the growth of other viruses. Okay, so these are some of the disadvantages of embryonated eggs. Then next one is cultivation, that is third method is tissue culture. Okay, the first application of tissue culture in virology was done in 1930, in which the rabbit was used, means rabbit vaccinia virus was mainly growth with the help of this tissue culture. As the name suggests, tissue culture means the tissues is isolated from various animals, and that tissue is mainly used for growth of the viruses. It is mainly of three types: organ culture, explant culture, and cell culture. Organ cultures are useful for isolation of some viruses which appear to be highly specialized parasites of certain organs. I mean, in this case, the particular organ is isolated from the animal, and into that organ, the viral suspension is inoculated. Okay. For example, tracheal ring organ culture is mainly employed for isolation of coronavirus, but that is not mainly used or generally used. Then explant culture. Explant means part of the organ which is mainly obtained from that organ, which is separated from the organ, and in that plant, in that part, the suspension of viruses is mainly inoculated. Minced tissue may be grown as explant embedded in plasma clots, but it is actually not useful into the virology. Then most popular and useful technique is cell culture. Okay, in this technique, the tissues are dissociated into component cells by the action of proteolytic enzymes such as trypsin and mechanical shaking. You can see into the image. A tissue is treated with enzymes. First step is tissue is treated with enzymes to separate the cells. Okay, so to these tissues there is a treatment has been given. So proteolytic enzymes, proteolytic as the name suggests, which cause the lysis of proteins with the help of enzyme trypsin and mechanical shaking. Okay, so likewise tissues are mainly dissociated into the cells. Okay, into the next step, the cells are washed, counted and suspended into the culture medium, and then it is distributed into petri plates, test tubes as well as bottles. Okay, the normal cells or primary cells grow in monolayer across the plastic or glass container. The transformed cells or continuous cell cultures do not grow in monolayer. Into the third step, you can see there are two kind of cells: normal cells and transformed cells. Transformed cell means in which the virus is mainly grown, in which the multiplication of viruses has taken place. Okay, so these transformed cells do not grow into the layer. Okay, while normal cells grow into the layer. Okay, so likewise we can easily identify these transformed cells, and these are mainly separated from the mixture. Okay, so likewise these steps are mainly done in case of cell culture. Then there are mainly three types of cellular culture. On the basis of the origin and characteristics, cell cultures are classified into three types, that is primary cell culture, diploid cell strains and continuous cell lines. Primary cell culture, these are normal cells freshly taken from the body and cultured. They are capable of only limited growth in a culture and cannot be maintained in serial culture. They are used for isolation and cultivation of viruses for vaccine production. Okay. Then diploid cell strains. Diploid cell strains are derived from primary cell cultures established from a particular type of tissue such as lung or kidney, which is embryonic in origin. They are of single type and can undergo 50 to 100 divisions before dying. They possess the normal diploid karyotype, that is set of chromosomes. As the name suggests, diploid means the two types of nucleus, that 2n type of nucleus is mainly present. They are mainly useful for isolation of fastidious pathogens and also for the production of viral vaccines. Okay. Then continuous cell lines. They are single type of cells mainly derived from cancer cells. These also can be grown in successive generations by transferring them from one test tube to another test tube without change in character of the cells. Okay. Virus growth in cell cultures can be detected by cytopathic effect, transformation, metabolic inhibition, interference, hemabsorption and immunofluorescence. Okay. So likewise, by using these techniques, the viruses are cultivated. So the cell culture, or you can say tissue culture, is one of the widely used method for cultivation of these viruses. Then we will see multiplication and reproduction of viruses. Multiplication, virus multiply, that is called as growth of virus. Okay, bacteria grow means bacteria multiply. So likewise, virus grow means virus multiply, and that can also be called as reproduction. Okay, virus mainly depends on synthetic machinery of host cell for replication because it lacks biosynthetic enzymes. Early studies on viral replication employed the bacteriophage as model. There are some of the similarities in the pattern of multiplication of bacterial and animal or human viruses. The steps of virus infection and replication can be divided into 6 sequential phases. Those phases are adsorption or attachment, penetration, uncoating, biosynthesis, maturation and release. We will see these steps one by one. First step is adsorption. The virus is adsorbed at a particular site on host cell, which is called as receptor. Into the image you can see the first step, virus, that is polyhedral virus has been shown, which has DNA, okay, and that mainly attaches to the host cell. Uh, then this virus come in contact with the host cell randomly, but this adsorption is specific and is mediated by binding of viral surface structures to the receptors on cell surface. So there is binding between the viral surface structure and the receptors. These receptors are mainly present onto the host cell, and this kind of binding is specific. For example, in case of influenza viruses, the hemagglutinin onto the virus gets attached to glycoprotein receptor sites onto the surface of respiratory epithelium. Okay, influenza is the infection of respiratory system, because into the respiratory system the receptors of influenza viruses are present. Okay, then in case of influenza virus, again they mainly attach to the glycoprotein, bind specifically to sialic acid residue onto the surface of respiratory epithelium. In case of HIV, that is human immunodeficiency virus, surface glycoprotein GP120 acts as the ligand. It mainly binds to CD4 receptors of T4 lymphocytes. Okay, so likewise this binding is particularly specific. So COVID-19 virus also has the receptors into the respiratory system, and that's why it mainly causes the infection related with the respiratory system. Okay, now the next step is penetration or entry of the attached virus inside the cell. There are 4 possible mechanisms which the virus use to enter into the host cell. Okay, for example, endocytosis, translocation, fusion, and direct fusion. You can see into the image. The non-enveloped viruses enters the cells by endocytosis or engulfment by invagination of a section of plasma membrane with accumulation of virus particles in the cytoplasmic vesicles. Okay, then there is another mechanism, that is translocation, which is the process by which the non-enveloped virus enters the host cell by moving across the cell membrane. Then fusion is the endocytosis of enveloped viruses, in which the envelopes of the virus fuse with the membrane of the endosome. And direct fusion of the virus envelope with the surface membrane of the cell also takes place in some of the families of viruses. Okay, so likewise into the second step, the virus penetrates or enters into the host cell. Then the next step is uncoating, that is the process. This is the process by which the virus loses its outer layer and the capsid. In some cases, uncoating is affected by lysosomal enzyme of the host cell. Okay, and there are other mechanisms by which the uncoating, means removal of protein coat or capsid from the virus, when it takes place. Then into the next step, there is a process of biosynthesis of viral components, in which there is synthesis of nucleic acid, capsid protein, and enzymes mainly takes place. Okay. The site of viral synthesis mainly depends upon the type of virus. DNA viruses synthesize their components in the host cell nucleus. Okay. Likewise, RNA viruses synthesize their components into the cytoplasm of host cell. Okay. Again, I will repeat. DNA viruses synthesize their components into the nucleus, while RNA viruses synthesize their components into the cytoplasm. Okay. Biosynthesis consists essentially of following steps. Those steps are transcription of mRNA from viral nucleic acid, then translation of mRNA into early protein. They initiate and maintain the synthesis of virus components and shut down the host protein and nucleic acid synthesis. Okay, viruses utilize the machinery of host cell, and that's why the protein as well as nucleic acid synthesis in case of host cell is only shut down. Okay, and that's why that host cell could not survive. That's why host cell is mainly died, and that's why the viruses mainly causes the infection, and that infection mainly spreads from one host cell to another host cell. Then there is replication of viral nucleic acid and synthesis of late proteins mainly takes place. The critical step in the biosynthesis is transcription of mRNA from viral nucleic acid. Once this is achieved, the host cell resources can be utilized for translating mRNA into viral components. Depending upon the structure of genome, viruses use different strategies for transcription of mRNA. Okay, so likewise, by the process of transcription, translation, and replication, as well as synthesis of various proteins, the biosynthesis of these components mainly takes place. Then the next step is fifth step is maturation, means assembly of nucleic acid and proteins. Assembly of virus mainly follows the synthesis of viral nucleic acid and protein. It may take place into the nucleus or cytoplasm. Okay, so if the virus has enveloped viruses get enveloped from the cell membrane of the host during the process of budding. Non-enveloped viruses are present intracellularly as fully developed viruses. Okay, then the last step is the release. In some of the cases, lysis of that particular host cell may take place, and those viruses which are multiplied, then they are released from the host cell, and from one virus, multiple copies of itself are mainly produced, and that's why it is mainly responsible to cause the infection. And this infection mainly spreads from one cell to another cells. Okay, and that's why these viruses are always infectious agents. Okay, so these were the steps which are mainly involved into the viral multiplication. Okay, thank you.